Well, first, I'd like to say thank you for all being here and good morning. I know it's early on a Saturday. So, for my Keystone project, I study chronic traumatic encephalopathy, or CTE. Some of you may have heard of this disease as it is commonly associated with high impact sports such as football or boxing. However, CTE has been seen in soldiers and construction workers as well. As CTE can only be diagnosed post-mortem, athletes go from being well-rounded stars to aggressive offenders for seemingly no reason. A notable case of this is that of Aaron Hernandez, who was an NFL star that became a suspect in a murder investigation as his behavior became increasingly erratic. After his untimely death, he donated his brain to the Anne McKee lab at BU. He was diagnosed with the worst case of CTE she had ever seen in someone his age. CTE is a neurodegenerative brain disorder, which progresses years after an individual's exposure to repetitive head trauma. The early stages, stages 1 and 2, are characterized by the onset of depression, emotional dysregulation, and aggression. In later stages, stages 3 and 4, memory loss and dementia are commonly seen. Aaron Hernandez had stage 3 and he was only 27 years old. People used to believe that concussions cause CTE, but it is actually years of microconcussions, which result in a prolonged inflammatory immune response in the brain. CTE is characterized by tau protein aggregation and neural atrophy. My project specifically focuses on neural atrophy and the inhibitory neuron populations in area 25 of the brain. This is a part of the medial prefrontal cortex, which has previously been associated with hyperactivity in depression, a major symptom seen in the early stages of CTE. I'd love to discuss my project more with you, so please come by and ask questions.
Good morning, everyone. My name is Gia Chavla, and I am a business administration major concentrating in entrepreneurship at the Questum School of Business. Before I start, I wanted to take the time to thank my professor, Peter Martin, for being such a good support system throughout this project. My project, The Startup Toolkit, is an ebook and podcast series designed to support emerging fashion startups in successfully launching their businesses by building a sustainable competitive advantage. As a fashion entrepreneur myself, I wanted to create a resource that would allow me to apply my own experiences while also learning more about the industry to understand what it takes to succeed as an emerging startup in the industry. The fashion industry is dynamic, fast-paced, and extremely competitive. This makes it difficult for emerging startups to enter the market and compete with established brands. These startups need to differentiate themselves and build an advantage that helps position themselves to stay competitive. To understand what it takes to identify and build this advantage, I focus my research around where the industry is and where it's heading. After a year of research, I identified 2 macro trends, sustainability and technology, that are shaping the future of this ever-evolving and fiercely competitive industry. And these form the core focus of the content in this guide. Specifically, how sustainability and technology can be drivers of this advantage needed to stay competitive in the industry. The Startup Toolkit provides a unique learning experience by combining audio and visual modalities. The guide takes a hands-on approach to understanding and applying the idea of building a unique advantage that can be sustained over time. Each podcast episode is accompanied by a set of Notion templates that include resources like case studies, frameworks, and strategies to help the reader understand and apply what they've learned. Additionally, the guide features a second brain master template that serves as an all-in-one virtual workspace to capture and organize ideas and track progress throughout the reader's entrepreneurial journey. Recognizing the importance of having a space for free thinking and reflection, this guide encourages you to brain dump your ideas and cultivate creativity and innovation. If you want to know more about my project and take a look at the actual guide, come find me at my poster. Thank you.
Hi, my name is Max Kroll, and my project was completed in the Zhang Lab on the Boston University Medical Campus. My project focuses on the interaction between the immune system and obesity, specifically in liver fibrosis. Liver fibrosis is one of the stages of liver damage, and it's categorized by a buildup of connective molecules and tissues in a way that eventually leads to scarring and inhibition of the underlying liver tissue from fully doing its job. It's well known that obesity is one of the leading causes of this. However, one of the more recent discoveries is that the immune system might play a role in causing liver fibrosis. And my lab studies how this happens at the cellular level. Specifically, we look at the role of neutrophils, which are a type of immune cell that usually works to kill foreign invaders, and how they might cause liver fibrosis. So to study this in my project, the first thing I did was I cultured liver cells in petri dishes. It was much easier for me to be able to control the variables in my experiment in a petri dish as opposed to using an animal model such as a mouse. What I would then do is I would treat one set of these cells with my enzyme of interest, which in my case was neutrophil elastase. It's the enzyme that neutrophils use to achieve a cellular response. And I would leave another set of these cells untreated. To observe the cellular response, I would use a process called immunostaining, which would allow me to tag certain markers of fibrosis, such as collagen, with fluorescent antibodies that could glow under a microscope and could illuminate whether a neutrophil elastase does have an effect on liver fibrosis. What I found was that neutrophil elastase does seem to have an effect on liver fibrosis, and thus inhibition of this enzyme is a potential drug target for reducing obesity-dependent liver fibrosis. However, my results were somewhat mixed, and further studies must be done to corroborate what I found using other biochemical methods. But please stop by my poster if you would like to learn more about the specifics.
Hey everyone, my name is Alyssa Goins, and the title of my project is Visualizing Anomaly Classification, Imaging Analysis for Landsat Data Archives. So for my project, I wrote the code for a computer software program that provides a visual interface for visualizing anomaly analysis data, specifically on Landsat imaging from the Amazon rainforest. So Landsat is a program launched by NASA in the 1970s that utilizes satellites equipped with sensors that take pictures of the Earth's surface. Anomalies in this visualization can occur when clouds and other objects obscure the land cover and don't allow the sensors to take proper imaging of the Earth's surface. This can present an especial challenge, especially when trying to analyze the imaging for deforestation patterns. For example, it would make it look like a tree did not exist when it in fact does exist on the land cover. So for my project specifically, I wanted to build a visualization software that would I wanted to build a visualization software that would provide immediate access to the imaging analysis that Professor Kastrian in the BU Math department is working on with his team. So I utilized MATLAB, and I essentially allow a user to upload a file directly to the interface. They're allowed to upload multiple files at once, allowing for simultaneous visualization. And the program will display the image in 3 panels. This is especially useful because in this type of research, one wants to examine multiple data points at once across the several different imaging. So this tool can be utilized in displaying and sharing data with a broader audience in order to share information on deforestation and climate change in the Amazon rainforest region. Thank you.
Good morning, everyone. My name is Julianna Helfer, and I'm a senior in the Human Physiology program at Sargent College of Health and Rehabilitation Sciences. For my Keystone project, I had the pleasure of working with Dr. O'Keefe, who is a professor at Sargent's Health Sciences program. So about 2 years ago, online, I took a sociology course through BU's Learn From Anywhere era, which we're all very familiar with. And I was introduced to the concept of social determinants of health, and I became very interested in looking at illnesses and health conditions through this lens. So social determinants of health are non-medical factors that influence our health outcomes. Most of the time when you're asked about your health, like at the doctor's office, you immediately just think of factors like your physiology, your genetics, maybe your family's medical history, but we often ignore those non-medical factors in those conditions in the environments that we live in, work in, go to school in, spend leisure time in, but they are just as significant to our health overall as well. So, for my project, I wanted to pick 2 specific conditions/illnesses that I was really interested in and are very relevant to our current time. So I chose COVID-19 and obesity. So for COVID, with the lockdowns of 2020, many low-income and marginalized communities were hit especially hard during the start of the pandemic and throughout the course of the pandemic due to health and social disparities that were already present beforehand. So things like being unsure about whether or not you'll be able to pay your rent, where your next meal will be coming from, if you'll be able to quarantine away from family members, and also if you'll be able to get tested for COVID-19 quickly and efficiently due to location and cost as well. Also, I wanted to focus on obesity because, as we all know, obesity is one of those pre-existing conditions, according to the CDC, that increases the risk of COVID-19 infection. And also, obesity in America is getting worse every year. The rates consistently continue to go up, and they are disproportionately affecting low-income and marginalized communities. So we're going through this COVID-19 pandemic while also simultaneously going through an obesity epidemic in America. So if you're interested in learning more about social determinants of health and how they intersect in terms of COVID-19 and obesity in the US, Massachusetts, and locally in Brookline and Brighton, then come visit me at my poster. Thank you.
Good morning, everyone. My name is Nicole, and my project is titled The Production of Central Tendency Effects in Interval Timing Tasks via Reinforcement Learning. And I've been doing this with Professor Ben Scott in the Department of Psychological and Brain Sciences. So in a nutshell, my project focuses on how the brain processes time intervals, specifically on the millisecond to second range. And that might seem very specific and very small, but it's actually something that plays into your daily life in a much bigger way than you would expect. For example, when you're driving a car, how do you know when to press down the brakes so you can reach the stoplight in time? Or even just making decisions. How much time is too much time spent on deciding on a decision versus how much is too little? So my project specifically looks at this through a task called the interval timing task, where participants are presented with a short time interval, usually through a light cue, and then they have to reproduce their estimation of that time by holding down a button for the length that they think that that time was. So during this task, a lot of the times there's this behavioral pattern that arises called the central tendency effect, where essentially participants will underestimate longer time intervals and overestimate shorter time intervals. And so we wanted to explore why this pattern tends to happen, specifically because in a lot of cases, people who are neurodivergent, so people with ADHD, people with autism, people with Parkinson's, there have been a lot of studies that show that these people actually do not exhibit central tendency effects, and they show very different behavioral patterns when they complete these tasks. So if we can kind of identify why they're happening in the neurotypical brain, we might be able to pinpoint places for treatment for these different types of disorders. So we do so with a number of computational models that focus on reinforcement learning algorithms, which if you would like to get into the specifics of that, please come visit me at my poster. I just want to end by thanking my advisor and everyone at the Scott Lab for all of their support.
Okay. Hi, everyone. My name is Olivia, and for my Keystone and Engineering Senior Design project, my advisor was Hannah Peterson. I partnered with her diagnostics company called Aperture Bio to develop a cytospinning apparatus for cancer diagnostics. So essentially, Aperture Bio collects biopsy samples from clinics, processes these samples, and analyzes images of them in order to detect the type of cancer that it is and the severity. In the processing part of this procedure, they do a technique called cytospinning, which allows them to concentrate cells onto a microscope slide for imaging. The current technology that they use for cytospinning is called the octospot, which is shown in that figure. The picture shows all of the components of the octospot, which get stacked together in the order as shown, put into that metal clip to make them secure, and put into the centrifuge for spinning. The process of spinning applies a force to the system that allows the cells that are in the 8-well strip to pass through the filter card and deposit onto the microscope slide. However, there are significant complications with the system as there are significant backorder issues with each of the components, which makes it inaccessible and unreliable. The outer 4 wells of the well strip do not function properly, which causes a 50% sample capacity loss. And the 8 well strip is actually a very thin one-use plastic, so it causes a lot of waste in this process. So our goal is to design, manufacture, and implement a new cytospinning apparatus to mitigate these complications. Our new device, as shown, consists of a well block, which was 3D printed using biocompatible filaments that are washable and reusable. It features 17 wells as opposed to the previous 8 and has alphabetical labeling of the wells for ease of use. We also made a corresponding filter paper using laser cutting, and all of this fits onto a standard microscope slide in order to make it integrated into any protocol. So we hope that our device allows Aperture Bio to improve their sample processing and get quicker results back to patients. Thank you.
Hi everyone, my name is Sophia Marcelli and my project was about emotional perception and subjectivity across the senses. So I'm actually going to pull up my experimental setup right away. What I wanted to do in this project was create a space where people could confront their own subjective emotional experience and also view that of others. What I ended up doing was I created 5 different artistic pieces and 5 accompanying music pieces, each corresponding with a different emotion, be that sadness, fear, or joy. What I asked participants to do was to come into the space and to place a sticky note on the floor. This sticky note should have been in a place where the volume from the music that was playing on the speaker and the ability to see the visual was essentially equal in terms of the emotional resonance that they felt from each component. Ultimately, what we found was that the data was really spread out. Every single person had a different experience, and I will have the pictures and audios as well as my results at my poster. But more than that, the point of this project was to create that space. And so it wasn't just about my understanding, but about other people's understandings of how they are different from the person next to them in terms of what they experience emotionally. I think that in a world where interactions tend to be a lot more contactless, understanding the subjectivity of something even as generic as enjoying a good piece of music is really important. And so I set out to, and I believe succeeded in creating a place where people could find that subjectivity. Thank you so much. I'd also like to thank my advisor, Professor Sam Ling in the Department of Psychological and Brain Sciences.